All right. Thank you all. It's been a great meeting. Really, a lot of really interesting, exciting talks. I want to thank Kenny and Ewan for this conference. It's been wonderful. Thank you for inviting us all and hosting us all. And I'm really excited about the session today, which is talking about ocular safety and some new findings with respect to the eye. So if I had to summarize my talk in one sentence, it would be, what is the role of the tear film in preventing far-UVC light-induced damage to any underlying ocular structures? But actually what I wrote in the abstract, and I think the take-home message, is that human tears provide little protection against far-UVC irradiation in human corneal epithelial cells. So the take-home message, despite what we've all been saying for the last 3 or 4 years, there is little protection of the tear film from far-UVC to the underlying structures. So therefore, I think we do need some more studies to understand the effect of far-UVC light on those underlying corneal structures. Another title for my talk might be Shedding Tears over far-UVC. I want to acknowledge my lab before I start. They're the ones who did all this work. Peter Arding will be talking right after me about some vision outcomes after far-UVC exposure in the murine model. And Imke Volkerts has 2 posters out there, which I hope you'll see about some other work we've been doing over the last year as well, and all the other members of my lab. So let's start with the human eye. This is a picture of the human eye. What we're mostly concerned with is the anterior surface, the light-facing surface of the eye. And you all know that the front surface, the clear portion of the front surface of the eye, is called the cornea. Cornea has an important function, but it's not the first surface that photons strike when they enter the eye, and that's the tear film. And we're concentrating on the tear film to a large degree during this session. It's a protective layer on the front of the eye. It's very thin. It's only 5 to 8 microns thick, but it is composed of 3 distinct layers. As Thomas said earlier, there's a deep layer, a mucin layer, which helps adhere the tear film to the corneal epithelium. The bulk of the tear film is the middle layer, the aqueous layer, and then up front is a lipid layer, which helps prevent dehydration from the surface. It's compressible, it's elastic, it clings to the surface of the cornea, and it has a fairly low protein concentration, only about 3 to 8 milligrams per ml, unlike a mammalian cell, which has a concentration in the range of 150 to 300 milligrams per ml. And predominantly the 3 main proteins found in tears are mucin, lysosome, and albumin, and some transferrin as well. What's the role of the tear film? Manyfold. It's a barrier. It protects us from the external environment. It has antimicrobial properties, the lysozyme and other elements, other proteins in the tear film help destroy bacteria that might invade the front of the surface of the eye. We have a cleansing function. We remove debris from the front of our eye, and that mucin layer helps prevent dehydration to the tear film and the underlying cornea as well. And when we blink, it provides lubrication to allow us to not damage the cornea as we blink. And we've all been saying that it also has far-UVC light absorbing properties. And that's what the purpose of this talk is today, to examine that in a little bit more detail. So again, a cartoon of the front of the eye, you can see 3 layers of the cornea with a lot of microvilli. So the reason the tear film provides an optical clarity to the eye is because those microvilli cause micro aberrations across the front surface of the cornea, and the tears help provide a single uniform lens and reduce spherical aberrations. The mucin layer adheres to the corneal epithelium, large aqueous layer, lipid layer on the front. Those are made by 3 different glands on the front surface of the eye. Peripheral to the cornea, we have the conjunctiva, and interspersed in the conjunctiva is a type of cell called a goblet cell. And goblet cells secrete mucin, which help adhere the tear film to the front of the cornea. As Thomas talked about earlier, the meibomian glands produce lipids, front surface of the tears, and the lacrimal glands produce the aqueous component of the tears as well. So showing histologically here, you see goblet cells interspersed with the conjunctival epithelium, and you can see in the picture above that the location of goblet cells peripheral to the cornea, the meibomian gland, and the lacrimal gland in the external adnexa. So what are the potential risks associated with far-UVC exposure? Damage to the corneal epithelium, to the corneal nerves that are posterior to that, damage to the conjunctival epithelium, or damage to the goblet cells, just to name a few. However, over the last 3 years, there have been quite a few papers that have suggested that the doses we're using at far-UVC, we're not seeing any damage to the eye. a number of experiments in both experimental human studies and in animal studies that suggest at the doses we're using, we're not detecting any kind of significant damage to the eye tissue. That may be in part because there's a constant renewal of the epithelium in the cornea. Shown in this picture, you can see there's a constant movement of the basal cells toward the surface, and those surface cells are shed fairly rapidly and discarded. So perhaps damage to those surface cells is not really relevant because they're shed and discarded. Furthermore, this has been done over the last 70 years, but Bodner is probably the classical demonstration of this. The penetration of UV light in the cornea is restricted, and 222 light is not getting very deep into the cornea at all. Excuse me. So back to our initial question, is far-UVC light absorbed by tears? We use two complementary approaches. First is physics and maths, spectrophotometry, and the Beer-Lambert law. The second is in the human corneal epithelial cell model, looking if tears or tear components actually prevent damage to the underlying epithelial cells. So let's talk a little bit about spectrometry and Beer's law. Very simple equation. The absorbance is a linear function of the molar absorptivity, which is a function of the specific proteins involved in a specific wavelength, times the path length that the light needs to move through, times the concentration of the solid, in this case proteins. So very simply, 222 light passing through a quartz cuvette of a defined path length. We measure the absorbance coming out of that cuvette. We know the concentration of what we're putting in that cuvette. We know that human tears contain a lot of macromolecules with a range of protein concentrations somewhere between 5 and 10 milligrams per ml, and that the majority of these are lysosome transferred serum albumin and mucin. And we know what the molar extinction coefficient is at 280 nanometers. That's well defined in the literature for lots of different proteins. Excuse me. So 3 different molar extinction coefficients for serum albumin, lysosome, transferrin, shown there. They're all relatively the same. So we can assume we can pick one of those to use for our model. And we know what the thickness of the tears are. Some variation depending on age, gender, pathology, but in general 5 to 8 microns. So using the Beer-Lambert law, at 280 nanometers, we can calculate what the transmittance is through the tears. And we see at 280 nanometers, we have very good transmittance, 99% transmittance. Sorry. Party last night was just too good. We can see we have very good transmittance at 280. What about 220? We can use the same calculation. We know that the tear film is 5 to 10 microns or 4 to 8 microns thick, so let's pick a 5 micron path length. We have to calculate the molar extinction coefficient at 220. We know what the absorbance is. We can measure that at 220. We know what the path length is, and we can look at the molar extinction coefficient, 531,440. Using that, we can back calculate the transmittance. So using a solution of serum albumin at a concentration relevant to that found in the tears, at a path length relevant to that of the tears, we have a transmittance of 91%. It's a model. It's not human tears. It's serum albumin at the concentration of protein found in human tears. What about real tears? Same calculation, using real human tears, our tears in our lab, using a different kind of an apparatus called a demountable cuvette. These are cuvettes that separate and come in different thicknesses. We're using a 10 micron cuvette, very thin, close to the thickness of the human tear film. We measure the absorbance using BSA or using human tears. You can see here we got a transmittance 83%. So I think using maths and physics, we've demonstrated that the transmittance of 222 far-UVC light through the tear film is at least 83%. So I think we answered the first part of the question. Second part of the question, can we see if relevant biological fluids or human tears prevent damage to underlying epithelial cells, human corneal epithelial cells? So we did first tear film protein measurements to make sure we knew what concentration of the protein was in the tears. In my case, I had to watch Marley and Me to get myself to cry a lot. Great movie, but very sad. Other people in the lab cut onions. Other people in the lab use something we use in Hollywood, which is a menthol stick, which makes you cry. And we got a fair amount of tears from members of the lab and other subjects. We measured protein concentrations. And in fact, the average concentration of protein in our tears is similar to that found in the literature. So somewhere between 3 and 5 milligrams per ml. We're using a human corneal epithelial cell culture It's called the ATCEPI. It looks very similar to normal corneal human epithelium. It has the same cell cycle kinetics, expresses the same proteins. It's a well-known tissue culture example for human corneal epithelium. And we use 3 assays for cell viability. MTT neutral red and lactate dehydrogenase release, which target 3 different parts of the cell. In the case of MTT, we're looking at mitochondrial function. A normal cell is going to convert a dye into a more colored dye that we can measure spectrophotometrically. A cell that is damaged to the mitochondria has a lower level of response. Excuse me. Neutral red is taken up by lysosomes. A healthy lysosome is a cell that's damaged, doesn't have much neutral red uptake, and a cell that's damaged at its membrane level is leaky, so lactate dehydrogenase leaves that cell into the external medium, and we can measure concentration in that way as well. So we measured viability through those 3 different measures. We looked at cell morphology, and we did this in multi-well plates. In a multi-well plate, the height of the medium, the smallest height of the medium that you can put over those cells is about 30 microliters, and the path length, the height for 30 microliters, is about 1 millimeter. But since there's a linear relationship between path length, concentration, absorbance, molar extinction coefficient, we can calculate what the equivalent concentration would be, and it's 0.05 milligrams per ml. So we plated human corneal epithelial cells. We exposed them to a fairly low level of light, 12 millijoules per centimeter squared, or 222 or 254, and other wavelengths. I'm only going to show you some of the data here because of time constraints. Different concentrations, different volumes. Here's an example of what we got. This is something Inka showed last year at ICFUST. This is without any tiers, just showing you the dose response to far-UVC in this cell line. You can see at fairly low exposures, above 3 millijoules per centimeter squared, we get a dose-dependent increase in the NPC, decrease in viability, decrease in MTT, decrease in absorbance of MTT. And when we do the same experiment with PBS and, excuse me, and BSA, we can see an equivalent concentration of 0.5 milligrams per mil, which is equivalent to the concentration of the tiers. We have very little protection in the MTT assay. You can see even at higher concentrations, we're getting very little protection at the equivalent depths. We did the same thing in that demountable cuvette I showed you I used spectrophotometrically. We did the same thing putting the demountable cuvette at a 10 micron path length over the cells. And you can see with human tiers or with BSA, this one's with BSA, I'm going to show you human tiers in just a second. Even at various dilutions, low dilutions, we're not getting any protection at all. So with 30 microliters of human tiers, same thing, multi-well plate, real tiers, no protection. And when we look at LDH release, similar kind of assay, human tiers as well, very little protection with even LDH release, which means the membrane is being damaged. And then in demountable cuvette, again, human tiers, no protection. So in conclusion, the relatively low protein concentration, the thin depth of the human tear film is inadequate to shield the underlying epithelial cells from damage induced by exposure to 222. I think I showed you that the Beer-Lambert law and symbol maths and physics show that at least 83% of incident light passes through the tear film. Using 3 different viability endpoints with either a proxy for tears, serum albumin or lactoferrin or transferrin solution, or actual human tears, we don't protect the underlying epithelial cells from damage. And that suggests we need to do a little bit more investigation into what's going on. So I love this quote, but when it comes to tears, I don't think we are shielding the underlying cells from harm. We need to investigate that a little bit more. However, as I said earlier, a great deal of data that suggests that in vivo, in the real world, with the concentrations we're using of far-UVC, we're not seeing damage. So we need to understand what's going on there. Last thing I want to say is that there have been some anecdotal suggestions. Some people have experimentally shown far-UVC lights in their eye and see some sort of stimulation activity or some sort of sensation activity. It could be this has to do with stimulation of the underlying corneal nerves. That's something we're interested in investigating a little bit more deeply. Thank you.